Hello everyone, welcome back to another session of regeneration biology. So in this class, we will learn a brief outline about spinal cord and brain regeneration and how different mechanisms comes into picture for regeneration of the brain and spinal cord. They are combined mainly because they are an extension. Basically, during development, the ectoderm forms a neural tube and give rise to a long neural tube, and the anteriormost portion of that neural tube expands and swells and becomes the brain. So basically, central nervous system ideally means brain and spinal cord. So hence the regeneration mechanism also have a lot in common, although the medical implication of their damage varies. Brain controls the entire body, whereas the spinal cord mainly controls the motor neuron functions of rest of the body like arms, body and legs, etc. So zebrafish and various other model organisms has been used to study the brain and the spinal cord regeneration, and we will see how and what are the approaches people take to study this. So to do brain injury studies, first thing is you need to know how a trauma is created, how a poking is created, and normally people use a mechanical injury method to both the brain and the spinal cord. You can poke a needle through the nostrils. That is basically the anteriormost part of the brain gets damaged. So you can see here there is a dot in the brain, and that is a place where you damaged it. And you can harvest the brain or you can section the brain, and you can take the images. And if you harvest the brain, you can get into RNA isolation, protein isolation. So this is the typical workflow that will be done, just like what you saw in the retina regeneration study also, where you do section and you take a selected block for visualization, etc. One such block of the cross section of the brain is what you are seeing. One is the left half, another is right half of the brain. So quiescent neural stem cells are contributing to the brain repair and regeneration. So keep in mind, every brain undergoes some small scale of damage, but many a times that will not reflect in loss of function, simply because for a given task or a given function, there will be more neurons allocated. Say for a job to be done, if 100 neurons are needed, it is not 100, there will be 10,000 neurons will be allocated, because of which even a few neurons are, I'm not talking about every function, a majority of the functions have got more neurons that are allocated. So even if some neurons are lost, then still that function will not be very badly affected. So although this is not a rule, but it is the way in which the organism survives. And in some organisms, there will be a persistent neurogenesis throughout the lifespan of the organism, because say for example, in zebrafish, the brain also keeps growing. Our brain doesn't keep growing. Your brain looks bigger simply because of the more and more neuronal connections are made. But the number of neurons are fixed in our case, in the case of humans. They don't increase in number because they don't proliferate. But newer and newer connections will be made, and which increases the bulk of the brain. So in adult mammalian brain harbors quiescent neural stem cells. They are called NSCs, which possess a latent capacity to generate neurons and glia. They have the ability, especially in the mice and rat, they have a zone called the subventricular zone, where there is a constant proliferation happens in the brain. Mechanistic, and that is lining the cerebral ventricles, the subventricular zone usually. Mechanistic insights into the origins and functional properties of quiescent NSCs starting to rise in rodents and Drosophila and various other regenerative vertebrates. People explore different animals based on the what angle you are looking into. Sometimes you may wonder why people are using different models. Why can't they stick with one model? So reason being, sometimes some model will be the best suited for a given set of studies, whereas other models are suited for another set of questions. That is why people explore different model organisms. It is becoming apparent that NSC, that is the neural stem cells, undergo different types of quiescence, such as G0, that is a G1 cell exit the cell cycle, that is called G0, and G2 quiescent, G2 cell exiting the cell cycle. So the normal cell cycle is G1, S, G2 and M. M stands for mitotic phase. Soon after mitosis, the cells are in G1 phase. Then if it decides to continue in cell cycle, it will go into S phase, that is the DNA replication phase. Once the DNA replication is perfectly completed, it will go into G2 phase. But cells have an option of getting into quiescence at G2 stage or G1 stage. So that is what G1 stage quiescence are usually referred to as G0 stage. Environmental signals such as exercise or feeding might increase the activation of the quiescent NSCs. You can have NSCs getting into activation. That means they can get more into a proliferative phase. Based on single cell transcriptomic data, putative trajectories from quiescence to activation, from dormancy to sleeping stage to wakeful state, that is called activation, have been reconstructed bioinformatically. People have explored those angles. In general, quiescent NSCs are restricted to producing specific neuron subtypes after activation in vivo. It is possible to modify these outputs experimentally in some instances. You can tweak the output. What are the possible cell type that is going to come from a particular activation event? The ability to control the outputs of quiescent NSCs will be an essential step in harnessing them for brain repair. Brain repair means brain regeneration. Those who are interested can read this Trends in Neuroscience article. So one factor that is contributing significantly to the brain regeneration is called BDNF. And zebrafish is a very well studied model organism to understand the brain regeneration. BDNF and brain regeneration and insights from zebrafish. Zebrafish is a teleost fish that is widely accepted as a model organism for neuro regeneration or neuroscientific studies. Adults show common vertebrate brain structures. Whatever a typical vertebrate, you take any vertebrate of your choice, including human, the typical structures are maintained. Although the zebrafish brain is small, human brain is big, but the typical structures are restored as it is in zebrafish, like that of in human. And they have similar key neuroanatomical and neurochemical pathways relevant to various human diseases. That is why we use zebrafish to study the regeneration biology. However, the brain of adult zebrafish possesses differences. Lot of differences are there from that of mammals. Intense neurogenic activity which can be correlated with high regenerative properties. This is something we should understand, that the neurogenic activity is quite intense. That means the zebrafish brain is waiting for an opportunity to make more neurons, that is neurogenic, pro-neurogenic. One such factor which is contributing or favoring is called brain-derived neurotropic factor. In short form, it is called BDNF. It is also important for the synaptic plasticity and maturation of neurons, etc. It is playing a vital role in various aspects of the brain function. It is a member of neurotropin family, has multiple roles in brain due to several biologically active isoforms interacting with different types of receptors. So BDNF is a well-studied molecule. Tropic factors means something acting on the surface. That is called a tropic factor. If you are applying, like you may have seen in ointment, tropical application only. That means on the surface. So BDNF is a tropical molecule. It's called a tropic factor. BDNF is a well-conserved vertebrate across millions of years of evolution, with the primary amino acid sequence between zebrafish and human BDNF 91% identical, not similarity. Identical means similarity will go much higher. Similarity basically means that basic amino acid or acidic amino acid, we call them homologous. But if it is identical, means it's not changing the amino acid at all. So 91% BDNF is identical. That tells you that no testing is done in evolution on that protein because it is playing a vital role. Same thing you can see about histone proteins also. Histone proteins are conserved from yeast to human. You can imagine yeast is a most primitive eukaryote which just start having the nucleus. Bacteria doesn't have histones. So that is also highly conserved protein. So BDNF is also highly conserved protein. The zebrafish brain is a valuable model to add new insights into the future of BDNF in regeneration studies. Brain-derived neurotropic factors and its mRNA distribution. So how regeneration studies are done? If you say, oh, gene A or gene B is important in regeneration, first thing is you should show a proof. After injury, what is its status? Does it come extra? Does it have a differential location or differential quantity of expression? And is the level going down, etc. So mRNA distribution is very vital in understanding. mRNA means protein also will be there. That is the assumption. And these are all done in the cross section. Different regions are there: telencephalon, mesencephalon, and this is the broad categorization: diencephalon, rhombencephalon, different regions of the brain. You don't need to worry about that anatomy part. All you understand is they are different extreme. Extreme is telencephalon, then mesencephalon, which is and then towards the spinal cord region, that is rhombencephalon, you can see. And normal injury will be done in the telencephalon. So the cells containing BDNF RNA, which is seen as red dots, because this is a cartoon, not real picture, but you can see here different areas there is red dots, you can see here, and also in this part. And then there is something called PCNA, proliferating cell nuclear antigen, which is indicative of that cell is proliferating. And then HUE. HUE is a freshly formed neuron, that they will have a marker called HUE. And this BDNF mRNA are present normally in the mature neurons. In several areas, they are close to the radial glial, that is the black dots. What you are seeing black dots is the radial glial cells, which has got the marker aromatase. And proliferating cell, green dots, which has got the PCNA marker. And the Q positive, azure dot, they are normally young neurons. You can see them in certain regions. The red dots, which is BDNF positive, and also the green, which is proliferating cell, they all converge, merge together. And the HUE cell, because normally if they are proliferating, that is also giving rise to new neurons. That is why the blue marker. And also you can see there is a supporting cell, that is the radial glial cell, which is seen as aromatase B enzyme positive. And the abbreviations are given for different regions, that is cerebellar body, dorsal telencephalon. So you can read it. It is not important, just for understanding. But this picture is put in just to show that the expression of BDNF have a strong correlation with proliferation and also formation of new neurons. That is the whole message that is given from this image. And if you look further, the BDNF in the brain regeneration, you can see here there is a telencephalic injury given using a needle. This picture we already saw it. And you can see as the days pass, 1 dpi, 4 dpi, 7 dpi, 15 dpi, and it continues. It takes for complete regeneration can take up to 6 months, but the studies are normally done up to around 2 weeks, because whether it is regenerating, the story will be complete by around 2 weeks of studies. And interesting thing is BDNF mRNA distribution in cross sections can be done at various time post injury. So you can see this width of BDNF is highest here, and by around 15 days it has become a narrow. So this means the expression, the spread of expression starts with a lot, as you can see here, plenty, because this is an injured area. There is a plenty BDNF expression, and it becomes less, and by 15 days it becomes very much restricted to the very close to the injury spot. So BDNF mRNA distribution in cross sections of zebrafish, telencephalon after injury. Cells containing BDNF mRNA, they are shown in red color dots, and also in young, HUE positive, and mature. And mature neurons have got acetylated tubulin. That marker is indicator of a mature neuron, whereas HUE marker, HUE expression is a neuron only, but it is young neuron, and they are shown in blue dots. So acetylated tubulin is old neuron or mature neuron, that is having blue dots, and the HUE positive, they are being given a blue color, that is freshly formed neuron. and in several areas they resulted in close to the proliferating. So the message is the newly formed neurons are seen very much close to the BDNF positive cells. So it indicates that the expression of BDNF is on a widespread area to start with, and that is indicative of PCNA, which is the indicator of proliferation, and also it has got BDNF and hue expressing co-expressing cell. BDNF and co-expressing cells are given like a circle label, you can see here, because the hue is blue inside and the red is outside, because it's a cartoon. That's how they have depicted. So many hue expressing cells have got BDNF also. So the take home message from this picture is the expression of BDNF is very high soon after injury, and as the regeneration gets completed, the BDNF level goes down and down, but the expression of BDNF is associated with the proliferation and also newly formed neuron. So these things tells you that BDNF is a pro-regenerative or pro-proliferative marker. And if you get rid of BDNF, you do not get normal regeneration. And you can see a schematic representation of adult neurogenesis, a comparison of neural stem cell niche to the telencephalic ventricular zone of zebrafish and mouse. So now in this picture, it's a cartoon where it is compared that how a zebrafish brain behaves versus a mouse brain behaves after an insult. So A is basically transverse section, that is zebrafish, transverse section through the telencephalon of adult zebrafish, illustrating different cell types present within this niche. A dash is the panel depicts the differentiation of radial glial cells. In short form, it is written as RGC into neurons and some signaling pathways controlling the activity of RGC, which happens in zebrafish. In zebrafish, quiescent RGCs, that is type 1, are activated to become the proliferative RGCs, that are type 2, which can regenerate proliferative neuroblast, that is type 3. So type 1 becomes type 2 and later type 3, eventually differentiating into mature neurons. So type 1 is basically a quiescent and becomes type 2 becomes proliferative, type 3 becomes neuroblast, that is capable of giving rise to other neurons. Transverse sections, that is the B panel in the mice, through the telencephalic region of the adult mouse. B dash shows the panel that is representing various steps of neural regeneration, starting from slow dividing neural cells, that is NSCs. Their rate of division is milder compared to that of zebrafish, referred to as B cells. The B cells give rise to C cells corresponding to highly proliferative progenitors, also called transiently amplifying progenitors or TAPs. The C cells differentiate into neuroblast, that is A cells in mice, and subsequently mature into neurons. So this is the pathway which is seen in mouse. Not that, but there is a difference. You may wonder, oh, both cases there is a proliferation, there is a neuron formation, so why not mouse also regenerate? So there is a catch. Let us see that. Not that the ventricular zone of zebrafish telencephalon, that is RGCs, correspond to the NSCs. Also, the neurogenic niches of the highlighted red color, as you can see here, highlighted red color in mammals, that is ventricular subventricular zone, that is VSVZ, and zebrafish is referred to as VC. Note that in mouse the NSCs are astrocytes, while in zebrafish NSCs are RGC, that is the radial glial cells. Although these two glial cell populations have different anatomies, they share very similar functions and genetic markers also similar in the ventricular zone of telencephalon. So you can see here the quiescent type 1 becomes type 2 and type 2 becomes type 3. Type 3 gives rise to the neuron. Whereas in B, the B cells, and because of the notch signaling, they can give rise to a transiently amplifying population called TAPs, and they give rise to A cells, which is similar to that of these proliferating cells, what you see in the neuroblast, similar to what you see in the zebrafish, and they neuroblast give rise to neurons. So this is a normal scenario which happens in both, but The regeneration context is slightly different. Activation of cellular and molecular processes upon injury in telencephalon, when you compare it, you will see the difference. In zebrafish, so this you are seeing it same zebrafish and mouse. Left, this is zebrafish top panel and the bottom panel is mouse. Both zebrafish and mouse, injury area is given capital A small a and capital B small a. AA and BA leads to apoptosis and necrosis in zebrafish. As a consequence, inflammatory signals are activated in zebrafish, recruiting immune cells such as neutrophils, microglia, and macrophages to the injury site, which you can see in AC and BC. In this picture, like you can see very clearly how injury causes the recruitment of various immune cells. In zebrafish, the activation of immune cells and inflammation is transient. Note that point very clearly, that inflammation you do not want persistently. You want it, you can't skip it. You want inflammation, but it is transiently and plays a positive role in activating RGC, that is the radial glial cells, leading to neurogenesis without scar formation. What is scar? Scar is nothing but ECM, extracellular matrix, especially collagen, which is produced by fibroblast and also various other immune cells. These cells, when they are produced, they will create a havoc. They are protective in role, but protection becomes so much that the cells don't have space to occupy in place where it should be there. Say for example, you are a furniture fan. Even a simple example, you like furnitures. Or one chair you bought, two chair you bought, three chair you bought, and you want to sit differentially. And now your whole house is full of furniture. Now, how people will come and move around in the house? Your house is only furniture, one top of the other, but because you like it. Furniture is needed, otherwise you can't sit on the floor. Same way, cells are attached to the ECM. They want to hold to the ECM. But the production of ECM becomes so high that will automatically lead to a condition called fibrosis, and there will be scar that will be formed in the lead without, and forming without scar formation is the beauty of regeneration in zebrafish. However, in mouse, let us see what happens. Inflammation leads to astrocyte proliferation. It is not like RGC in the case of zebrafish. Astrocyte proliferation, and in most cases, astrocytes are involved in glial scar formation. Additionally, extracellular matrix deposition impedes, not the word impedes, neurogenesis and neurite plasticity, because there is lot of blockade. It is just like traveling, taking your cycle through a very crowded road, a traffic jam road. You are going in bicycle, even walking by foot, it will be very difficult. So you have too much of junk that is formed, which will prevent the neurogenesis, formation of new neurons, and the neurite plasticity, making new connections, hindering successful regeneration. So the formation of too much of ECM hinders the successful regeneration. Unlike in zebrafish, not that point also, unlike in zebrafish, in both models, there is an observed increase in oligodendrocyte, which is a central nervous system specific glial cell, oligodendrocyte recruitment and their proliferation. Still, this recruitment or proliferation is much more pronounced in mouse, more than required, one can say, more than required response than in zebrafish. Not that the direct experimental evidence about the recruitment of peripheral immune cells to the injury site of adult zebrafish telencephalon is lacking. That means there is not too much of aggressive recruitment of immune cells in zebrafish, because of which the immune response is minimal, and hence the ECM or the scar formation is minimal. Hence the newly formed neurons get a chance. It will get a chance to migrate effectively and form newer connections. So the details are, those who are interested can read this paper, and the details are well described in the image. And if you look the other part, this topic is also about spinal cord regeneration. Spinal cord injury is one of the most prevalent example of roadside accident. People get spinal cord injury in road accident and they get paralyzed below their waist. typically results in axonal damage and death of the neurons and glial cells, which results in paralysis in the case of humans and any other animal. Spinal cord is very important part of the central nervous system. The secondary injury phase is primarily caused by uncontrolled inflammation. Remember, inflammation came once again. Excitotoxicity, edema, ischemia, and chronic demyelination. Myelin means basically a covering of your neurons. Demyelination means something like a multiple sclerosis condition where signal will get leaked out. So brain is sending signal, but it is not reaching the target. In between, it got leaked out. So demyelination causes that at the injury site. Glial scars are generated at the wound site, can inhibit axonal regeneration. Same issue what we discussed a while ago. Too much of scar formation prevents the prospect of regeneration. So, comprehensive illustration of 6 major challenges in SCA, spinal cord injury treatment. Challenge 1. Unclear cell types. What are the cell types? Unclear nerve regeneration cell types limit the stem cell therapy because some junk gets filled in that place. Challenge 2. Abnormal microenvironment. Injury site conditions impair transplanted survival, cell survival and its function. So even if you put stem cell, it doesn't help much. Inhibitory immune response. So prolonged dysregulated immune response hinders nerve regeneration. And challenge 4, glial scarring, reactive astrocytes from the barriers inhibiting axonal regeneration. That is another trouble. As you can see here, glial scars are being formed here. And challenge 5, bridging large gaps. That is, after spinal cord injury, the spinal cord is separated, this much gap is there, creates gaps, challenging the axonal regeneration in an unknown territory. Like if you have one building and two building, if it is 1 meter distance, you will jump. If one building and second building have got 10 meter, how will you travel? You can't, no scope of jumping from that. Same logic applies here. Axonal regeneration and neuronal connections. Challenge 6. Limited functional recovery. Functional, even though mechanically you are linking, functionally they are not the same. Functional recovery is limited due to axonal integration challenges. So these are all the main challenges what has been faced by comprehensive spinal cord injury response. A comprehensive schematic of zebrafish experimental model utilized for system of regeneration. So how people do, zebrafish does this very effectively. So starting from zygote, if you see, zebrafish is vulnerable for transgenesis, genome editing, etc. You can use either juvenile zebrafish or even use embryos itself. 72-hour post-fertilization embryo people use for spinal cord injury. And SCI approaches can be mechanical damage, and it can be a stab wound or a scissor cut wound. And you can also isolate number of regenerative cells that is responding from the young embryos much easily because the tissues are not very harsh. And also people do cell transplantation therapy. You can take the cells, whichever cells, and you can put in that place and see what happens to the fate of those cells. Do they transdifferentiate or they differentiate into the neurons? So that is transplantation therapy is possible. And the introduction of regenerative cells into the lesion, and they can stimulate the regeneration and the recovery. So these are all the approaches that is normally done using zebrafish as a excellent model. Coming back to this point, we should understand that every situation, that each challenges we saw, 6 challenges we saw, each challenges comes with its own baggage. Like inflammation creates lots of free radicals, and although inflammation is needed for the initial immune response, but these free radicals can cause cell death also. So even if a injured spinal cord thinks of regeneration, but the ROS formed can inhibit their normal survival. And also there are several axonal growth inhibitors. Central nervous system do not want any new communication, because any new communication is the stored information will be altered. So these inhibitors has to be removed, so that they should be told, oh, now regeneration has to happen. So let us keep away from these troubles. So we will study about regeneration biology in the next class. Thank you.